Hello everyone. In this class, we are going to learn about the erythropoiesis, the mechanism by which the RBCs are produced in bone marrow. So this process is called as erythropoiesis. The RBCs have a lifespan about 120 days. The bone marrow precisely replaces the cells lost by senescence, hemorrhage or destruction. Red mass cell in normal men is around 26 to 32 ml per kg, and in women it is 23 to 29 ml per kg body weight. Sites and stages of erythropoiesis. By definition, erythropoiesis is defined as the process of formation of red cells. So this is important component of hemopoiesis. So there are three stages of erythropoiesis: mesoblastic stage, hepatic stage, and medullary stage. Mesoblastic stage, during intrauterine life, erythropoiesis first takes place in the mesoderm yolk sac and mesoderm of the body. So this is called as mesoblastic stage. During this stage, erythropoiesis is intravascular. Hepatic stage occurs from the 5th week of gestation. Erythropoiesis takes place in the liver and spleen. Medullary stage, from 5th month of intrauterine life, the bone marrow starts forming red cells. However, erythropoiesis in bone marrow is very slow in second trimester. Medullary hemopoiesis becomes more effective towards the end of third trimester. After birth, bone marrow becomes the sole site for erythropoiesis. Extramedullary erythropoiesis in postnatal life is always considered as abnormal. Till adolescent period, the marrow cavity of all the bones are involved in erythropoiesis, after which erythropoiesis regresses in limb bones. After the age of 20 to 30 years, erythropoiesis is mostly limited to sternum, ribs, vertebrae, skull, pelvic and pectoral girdles. Steps of erythropoiesis. So there are four major cell stages of steps of erythropoiesis: stem cells, progenitor cells, precursor cells, and mature cells. In each stage has substages. The red cells are formed from the pluripotent hemopoietic stem cells, which give rise to committed stem cells. Committed stem cells form a subsequent progenitor and precursor cells that finally produce reticulocyte and mature red cells. In general, during erythropoiesis, following cellular changes takes place. So these are important. Cell size progressively reduces. The size of the nucleus and number of nucleoli decreases. Chromatin material condenses, and finally nucleus disappears. Staining reaction of cytoplasm changes from deep basophilic to polychromatophillic, that is acidophilic plus basophilic, and finally acidophilic type. This occurs mainly due to the gradual reduction in quantity of RNA material in the RBC. Pluripotent stem cells, these are mother stem cells and that form stem cell for different cell lines. Stem cells, which including committed stem cells, have two unique properties. One is self-renewal and they have ability of differentiation. They have a capacity to renew themselves, therefore at no time bone marrow is depleted from stem cells. They also have a capacity to develop or differentiate into subsequent progenitor cells. Committed stem cells, they are derived from pluripotent stem cells. There are specific two categories of stem cells for myeloid series and lymphoid series. Myeloid stem cells form erythroid series, megakaryocyte series or monocytic series and granulocytic series. Erythroid stem cells give rise to progenitor cells and erythroid cell lines. So there are two progenitor cells which are called as BFU erythroid series and CFU erythroid series. As I discussed, there are two types of progenitor cells, BFU-E and CFU-E. The BFU-E and CFU-E develop from the common progenitor, that is CFU, uh, megakaryocyte and erythrocyte series. BFUs-E are the burst-forming units of erythroid progenitor cells that give rise to large number of CFU erythroid cells. And CFUs-E are the colony-forming unit erythroid progenitor cells that give rise to moderate number of blast cells of erythrocytic series. Thus, BFU-E forms CFU-E, which in turn forms the blast cells, which are called as erythroblasts. As blast cells are first to be morphologically identifiable cells in the bone marrow during erythropoiesis, they are the true precursors of erythrocytic series. The precursors of erythrocytes are called as erythroblasts, and also called as Normoblast. Nommoblast developed from Pronormoblast. There are three successive forms of these Nommoblasts: early, intermediate, and late forms. Pronormoblasts are the first blast cell to appear in bone marrow and the first identifiable cells in the erythrocyte series. The following are the characteristics of a pronormoblast. So these cells are large, having diameter of 15 to 20 microns. Cells are irregularly rounded or slightly oval. The cytoplasm is less in amount, occupies around 20% of the cell. Presence of high concentration of polyribosomes makes the cytoplasm intensely basophilic. Cytoplasm has high RNA content, which indicates vigorous protein synthesis in the cell. The nucleus is large and occupies almost 80% of the cell. It contains multiple nuclei. Mitosis is present. Hemoglobin is not yet formed in the cell in pronormoblast. In early normoblast, which is also called as basophilic erythroblast, is the first erythroblast to appear in the bone marrow. So the features are diameter is around 12 to 18 microns. They exhibit active mitosis. Cytoplasm is scanty, deep blue, and basophilic. The basophilic cytoplasm is due to continuation of many polyribosomes in it. Therefore, the early normoblast is also called as basophilic erythroblasts. Nucleus is large and occupies almost 3/4 of the cell area. The nucleus is composed of dark violet heterochromatin clumps interspersed with a pink clumps called as euchromatin. The chromatins are connected by linear strands, so this often gives a nucleus the appearance of wheel spokes. So hemoglobin appears first time in these cells during erythropoiesis. Intermediate normoblast. So this is also called as intermediate or polychromatic erythroblast. So this appears following mitotic division of early erythroblast. Following are the features of intermediate erythroblast. The cell is smaller than the early normoblast, having diameter of 10 to 15 microns. The cytoplasm changes from blue to pink as hemoglobin dilutes the polyribosome content. Nucleus is small and occupies about half the cell area. The distribution of heterochromatin clumps in nucleus gives appearance of checkerboard pattern. So there are no nuclei in intermediate normoblast. Hemoglobin synthesis increases, which makes the cell more acidophilic. The presence of RNA material makes the cytoplasmic eosinophilic. So thus, mixture of acidophilic hemoglobin and eosinophilic RNA in cytoplasm makes it polychromatic. So that is why the intermediate normoblast is also called as polychromatic erythroblast. Late normoblast. This is also called as, this is produced by the final mitosis in the erythropoietic series. Features are the cells are 7 to 12 microns in diameter and mitosis is absent. Cytoplasm is eosinophilic, deeply eosinophilic, giving appearance of orthochromatic cells. So hence it is also called as orthochromatic erythroblast. Nucleus here is small, pycnotic, and uh, pycnotic at the beginning, sometimes dark nuclear chromatin materials are arranged in a typical pattern to give the appearance of cartwheel. Finally, the pycnotic nucleus disintegrates. Hemoglobin synthesis increases and almost completes at this stage. Mature cells are called as reticulocytes. So reticulocytes are the immediate precursors of red cells. Therefore, they are also called as juvenile red cells. They are mature cells with a full uh, complement of hemoglobin. They are slightly larger than red cells. They have a network of reticular nuclear material. The reticular network is nothing but the remnants of disintegrated organelles and especially of the nuclear fragments. Due to the presence of reticular network, the cells are called as reticulocytes. The reticulum is stained by supravital stains. Hemoglobin synthesis continues to some extent in some reticulocyte. The reticulocyte count is around 0 to 1% uh, of red cells in adults. Erythrocytes are the final cells of erythropoiesis. They are biconcave disc and they have a diameter of 7.5 microns. Okay, so these are the different stages of erythropoiesis. Okay, so from the hematopoietic stem cell to the myeloid progenitor, CFU, uh, megakaryocyte erythroid series, BFU, CFU of erythrocyte series, Pronormoblast, uh, Basophilic early Nommoblast, intermediate Nommoblast, late Normoblast, reticulocyte, and the mature RBC. The total period for erythropoiesis is around 7 to 9 days. So it takes 5 to 7 days for progenitor cells to become reticulocyte and another 2 days for reticulocytes to become mature red cells. How the erythropoiesis is regulated? So there are many factors but that they are effectively controlled. In physiological condition, total cell mass is maintained at its optimal size by suitable adjustment in the rate of erythropoiesis. There are various factors which are controlling erythropoiesis. You can divide them into the humoral factors, dietary factors, and other factors. Okay. We will be discussing them in one by one. Okay. So in animals, spleen is the reservoir for the red cells, but not in humans. There are no specific origin of signals from particular reservoir for the production of red cells, and consequently there is no anatomical tissue feedback for erythropoiesis. The mechanisms of controlling erythropoiesis can be broadly divided into two categories: the functional feedback and the end product feedback. Functional feedback is the feedback that originates from the tissues, signaling the rate of their requirement, which normally is served by the red cells. Red cells supply oxygen to the tissues, and therefore, oxygen requirement of the tissue is the major functional feedback for the red cell production. And erythropoietin is the chief mediator of this functional feedback. End product feedback. The end product feedback is due to the products of red cell destruction. It is believed that the products released from red cell destruction influence the red cell production. This is supported by the observation that hemolytic anemia is associated with more erythroid hyperplasia and reticulocytosis than the hemorrhagic anemia of equal severity. The precise mediator of end product feedback is not clearly identified, though in vitro studies have demonstrated that the stimulatory effect of hemin on erythropoiesis. So factors those control erythropoiesis can be broadly divided into hormonal and dietary and others. In hormonal factors, important is erythropoietin. Okay. So French professor Paul Queneau and his associates suggested that hypoxia generates humoral factor capable of stimulating red cell production. In 1950, Reisman provided strong support evidence for existence of hormonal mechanism, and few years later it was named as erythropoietin. Jacobson and co-workers found that erythropoietin is produced by the kidney in 1957. Normally, erythropoietin is produced by mainly the interstitial cells in peritubular capillary bed of the kidney. To some extent, it is also produced by juxtaglomerular cells and the extraglomerular mesangial cells of the kidney. Kidney secretes about 85% of the erythropoietin and rests from the liver. Non-parenchymal cells in the liver and perivenous hepatocytes, they also produce erythropoietin. There are also evidences that erythropoietin is produced from brain, uterus, and oviducts. Erythropoietin acts on erythropoietin receptors that belong to cytokine receptor superfamily. The receptor has tyrosine kinase activity. Binding of erythropoietin with its receptors initiates activation of cascade of serine and threonine kinases that finally leads to activation of JAK2 protein. Formation of JAK and other signaling proteins bring about the changes in the cell function in a similar way to that of growth hormone. Functions of erythropoietin: it stimulates the erythropoiesis in several ways. It acts mainly on the progenitor cells and early precursor cells, especially stimulates BFU-E and CFU-E to form Pronormoblast. It decreases the cell cycle length of precursor cells and therefore enhances mitosis. It facilitates the maturation of Nommoblast. It increases the hemoglobin synthesis in Normoblast. It acts on the stem cells to promote their transformation towards Erythroid series. It stimulates early release of immature erythrocyte, that is reticulocytes, into the circulation. How erythropoietin production is regulated? Okay. So the factors which regulate the EP production can be divided into factors increasing and factors decreasing the production. The factors which increase erythropoietin production are hypoxia, low blood volume, anemia, lung diseases, and hormones like epinephrine, norepinephrine, androgen, thyroxine, prolactin, ACTH. Okay. And factors that decrease erythropoietin production are estrogen and adenosine antagonists such as theophylline, okay, so which are going to decrease the erythropoietin secretion. So metabolism erythropoietin occurs in the liver, usually inactivated in the liver. The half-life of erythropoietin is around 5 hours in the blood. If the carbohydrate component, especially the sialic acid residue of erythropoietin molecule, is damaged, the half-life can be shortened to 5 minutes. Interleukin and GM-CSF, the interleukin 1, 3, 5 are produced mostly from the T cells, act mainly on the stem cells to convert them to the progenitor cells. GM-CSF, which is produced from T cells, endothelial cells, and fibroblasts, stimulates the production of committed stem cells. Androgens stimulate erythropoiesis. So this is the major cause of higher red cell count in males than in females. Red cell count is same in both genders till puberty. Androgen stimulates erythropoietin production and directly stimulates erythropoiesis. Estrogens inhibit erythropoiesis. Estrogen inhibits erythropoiesis by inhibiting erythropoietin production and also by decreasing the response of stem cells to erythropoietin. So it also decreases hepatic synthesis of globulins. Thyroxine stimulates erythropoiesis. Though the exact mechanism of thyroxine increasing the red cell count is not known, but it stimulates erythropoietin production. Growth hormone increases the mitosis and maturation of erythroid precursors. Cortisol produces mild erythrocytosis. Iron is the raw material for synthesis of heme component of hemoglobin. So it almost occupies 35% of the RBC. Therefore, iron deficiency results in hypochromic microcytic anemia. Vitamin B12 and folic acid are the vitamins necessary for maturation of red cells and precursors as they promote DNA synthesis. For synthesis of DNA, thymine is required and tetrahydrofolate is necessary for thymine synthesis, which is formed from the folic acid. Folate after its reabsorption from the intestine becomes methyl tetrahydrofolate in blood, which is the which in the tissue becomes 5,10 methylene tetrahydrofolate, the active form of tetrahydrofolate. This promotes the formation of deoxythymidylate from the deoxyuridylate, which is required for the synthesis of DNA. Therefore, folate deficiency lead to arrest of mitosis and chromosome division in the absence of DNA synthesis. Here in the figure you can see that the folate methyl tetrahydrofolate is a dietary uh, folic acid which gets converted into tetrahydrofolate with the effect of with the presence of vitamin B12. Okay, and this tetrahydrofolate is in the tissue gets converted into 5,10 methylene tetrahydrofolate, which is important for the DNA synthesis. Okay, and the deficiency of both dietary folate and vitamin B12 can lead to reduced maturation of the RPC. So vitamin B12 promotes the conversion of methyl tetrahydrofolate to its active tetrahydrofolate form. The methyl tetrahydrofolate THF accumulates in the cell, which is known as methyl THF trap. Therefore, vitamin B12 deficiency also causes arrest of chromosomal division. In fact, folic acid and vitamin B12 interact with each other for the synthesis of DNA. So thus in deficiencies of these two vitamins, megaloblasts are produced in the bone marrow instead of normoblasts, therefore resulting anemia, which is called as megaloblastic anemia. So they are the macrocytes and it is also known as macrocytic anemia. Okay, so these are the diagrams of macrocytes in folate or vitamin B12 deficiency. Protein is also essential for synthesis of globin component of the hemoglobin. Therefore, protein deficiency is invariably associated with hypochromic anemia. So other nutritional factors like vitamin C helps in absorption of iron. Therefore, vitamin C deficiency leads to anemia. Minerals like copper, cobalt influence hemoglobin formation. Thus, deficiency of these minerals leads to anemia. Other factors like intrinsic factor, which is secreted from the oxyntic cells of stomach along with hydrochloric acid. It helps in the absorption of vitamin B12, so which is nothing but extrinsic factor of the castle from terminal part of the ileum. Intrinsic factor deficiency produces megaloblastic anemia or pernicious anemia. Other environmental factors like hypoxia due to any cause, it increases the erythropoiesis by increasing erythropoietin production. The common physiological cause for hypoxia is high altitude. Hypoxia occurs at high altitude due to the decreased oxygen content that decreases the oxygen availability to the tissues. Pathological causes of hypoxia are anemia, low blood volume, cardiac diseases that decrease the tissue blood flow as it occurs in heart failure and respiratory diseases. Some vasoconstrictor drugs like catecholamines, nucleotides like cyclic AMP, NAD, NADP, products of red cell destruction, hemosolates, cobalt salts, and thyroxine, they are going to stimulate the erythropoiesis. Under normal conditions, most of the red cells are produced in the bone marrow and are actively alive or have the potential to live a normal lifespan. So this is called as effective erythropoiesis. A fraction of red cell production is ineffective with the destruction of non-viable red cells either within the marrow or shortly after they are released into the circulation. So when percentage of production of these non-viable red cells increased, so the erythropoiesis is called as or termed as ineffective erythropoiesis. The ineffective erythropoiesis is suspected when reticulocyte count is normal despite of erythroid's hyperplasia in marrow. So this is how hypoxia is going to regulate production of the RBC. Hypoxia leads to reduced tissue oxygenation that increases the erythropoietin production. Okay, so that is going to stimulate the hematopoietic stem cells to produce red blood cells, so which helps in the oxygenation of the tissue. So and that can act as a negative feedback mechanism for the production of erythropoietin in the kidney. Reticulocytes are also called as juvenile red cells in the bone marrow. They develop into mature red cells and few of them pass into circulation along with red cells. During the process of development of cell in the erythrocytic or erythrocyte series, the nuclei are lost but not the cytoplasmic RNA. Reticulocytes do not possess nuclei but contain network of reticulum in the cytoplasm that represents the remnants of basophilic cytoplasm, that is the RNA material of the precursor cells. So here these are the reticulocytes and these are the reticular network. On vital staining with cresol blue, the reticular network appear in the form of heavy wreath or clumps of small dots or as a faint threads connecting two small nodes. The ribosomal and cytoplasmic remnants of reticulocytes pick up supravital stain like a new methylene blue. The stain is allowed to penetrate the cells while in the living condition. Supravital stains may also reveal the basophilic stippling. Red cells are smaller than reticulocytes and they do not contain any nuclear material. Reticulocytes are produced in the bone marrow from late normoblast. Okay. The nucleus is removed from the late normoblast to become reticulocyte. Reticulocytes lose their mitochondria and ribosomes and basophilic tend to become mature red cells. About 1% of circulating red cell is replaced every day by newly formed red cells. With the release of young red cells into the circulation, few reticulocytes are also released. When the bone marrow sends out red cells at an increased rate, more reticulocytes are also released. So thus the number of reticulocytes in the peripheral blood is an index of erythropoiesis, especially in case of anemia. What is reticulocyte response? The reticulocyte count is performed to assess the therapeutic response of anemia to the treatment in which the patient is deficient in one or the other substrate essential for the synthesis of red cells. When therapy begins, new red cells are formed and released rapidly into the circulation before cells are fully mature. Therefore, many reticulocytes are also released with the release of young red cells. This results in increased reticulocyte, which is called as reticulocyte response. Reticulocyte response indicate a favorable response to the treatment. Especially, this is typically observed in beginning of the treatment for the pernicious anemia or iron deficiency anemia. When vitamin B12 is administered as a part of treatment for pernicious anemia, the number of reticulocytes increases in the blood in the initial phase of the treatment, which indicates that the patient is responding well to the treatment. When iron is given in the treatment of iron deficiency anemia, reticulocyte number increases. Okay. So these are some differences between reticulocyte and red cells. Obviously, reticular cells are a bit larger compared to the red cells. Reticulocytosis is increase in the reticulocyte count. So this is seen in various physiological and pathological conditions. Physiologically, in newborns and infants, okay, and there also count is more when you are in high altitude. Pathological causes are hemolytic anemia, acute hemorrhage during the treatment of deficiency anemia, and any condition that stimulates the bone marrow to produce red cells. Reticulocytopenia is a decrease in the count. There is no physiological reticulocytopenia. Reticulocytopenia is always indicates disease state. It is seen in aplastic anemia, myxedema, hypopatriotism, and leukoerythroblastic anemia. Leukoerythroblastic reaction is used to describe the presence of immature myeloid cells and nucleated red cells in the peripheral blood, which occurs often as a consequence of disturbance of bone marrow architecture by abnormal tissues. This is seen in secondary carcinoma of the bone, myelofibrosis, post-splenectomy states, thalassemia major, and multiple myeloma. Reticulocyte count remains normal in ineffective erythropoiesis. In fact, ineffective erythropoiesis is suspected when reticulocyte count is normal despite of erythroid hyperplasia in bone marrow. The effective erythropoiesis is estimated by reticulocyte count, which is normally expressed as a percentage of red cells. However, it can also be expressed as the total number of circulating reticulocytes per unit of blood, that is absolute reticulocytes is equal to reticulocyte percentage into red cell count. To use the reticulocyte percent as a measure of rate of red cell production, the percentage may be corrected for the hematocrit. So this is called as reticulocyte index or corrected reticulocyte count. Therefore, reticulocyte index is equal to reticulocyte in percentage into actual hematocrit divided by normal hematocrit. So this is all about in brief about the erythropoiesis in bone marrow.